to the Emery Medical Genetics review series. In this series, we make the concepts from Emery Medical Genetics 16th edition, which is the latest edition from Emery, clear and connected. Think of it as a roadmap. Instead of reading chapter by chapter, we bring together discussions scattered across the book into one integrated concept, so you see the whole picture at once. These videos are designed for biology undergraduates preparing for advanced studies, medical students mastering their core reference, and graduates or professionals refreshing their knowledge. You can use this series in different ways. Watch first for an overview, learn alongside the textbook, or return later for a quick review. Let's dive into today's concept. Hello everybody! Today we are going to dive into very foundational concepts in biology that are crucial for understanding how life works at the cellular level. The concepts that we are going to talk about are the central dogma, the structure of DNA, DNA replication, and the structure of chromosomes. First, we want to talk about the concept of the central dogma. Actually, it's the core principle that governs the flow of genetic information, which begins with the DNA. Then, through the process of transcription, we would have the RNA molecule. And then, through the process of translation, we will have the final product, which is the protein. This is the main flow of genetic information in our body. However, we have some exceptions about the central dogma. I have said that we have DNA. DNA is actually located safely inside the cell's nucleus, and DNA holds all the hereditary information needed to build and operate the entire organism. And this is actually the permanent set of recipes of whatever we actually require to perform as an organism. Next, through the process of transcription, we will have the RNA. Actually, the cell knows that it can't risk taking the entire DNA out of the nucleus. So instead, when it needs to make a specific product, it creates a temporary working copy of just the recipe, which is the gene actually it needs. So this copy would be made through the process of transcription, and it is a molecule called RNA. Then we would have the process of translation. The newly made RNA copy travels out of the nucleus into the cell's main workspace, which is the cytoplasm. Here, a cellular machine called a ribosome reads the RNA and assembles the final product, which is a protein. Actually, proteins are the hard-working molecules that carry out nearly all the main cell's functions. It is important to understand that this isn't a simple one-to-one conversion, and a single gene can actually give rise to many different proteins thanks to a complex network of regulatory steps, which we will discuss further. This is the reason we can have something as complex as the human brain despite having a relatively limited number of genes. Then we want to talk about the functional anatomy of the cell. The cell is the basic unit of life, and it's organized into specialized compartments called organelles. Actually, as you can see in this picture, the cell's common center is the nucleus, which contains the DNA and protects it with a membrane that has pores. These pores regulate what enters and exits, including our RNA copies. Surrounding the nucleus is the cytoplasm, a jelly-like substance where all the other organelles are suspended. Within the cytoplasm, we find a number of key players. The ribosome, for example, are the factories where translation occurs. To get the energy needed for all these tasks, the cell relies on its mitochondria, which is often called the powerhouse of the cell. Then there is the endoplasmic reticulum, which is called ER. It is a network of channels often associated with ribosomes, where proteins and lipids are synthesized. Once products are made, the Golgi apparatus acts like the cell's post office, actually, processing and packaging them for transport. So it would be very important for cell to package and process its final products through this important organelle called Golgi. Finally, organelles like lysosomes and peroxisomes serve as the cell's recycling center, which means that lysosome and peroxisome can break down waste materials and toxins. These organelles are not isolated. They are highly interconnected. Consider a protein's journey actually inside a cell. It begins at a gene in the nucleus, Then it is transcribed into RNA, which exits into the cytoplasm, is then translated by ribosomes on the ER. Then it will be modified and packaged by the Golgi, and then, if no longer needed, it would be sent to the lysosomes for disposal. All of these steps are powered by the mitochondria, which is the powerhouse of this cell. Then we will talk about the RNA, DNA, and RNA, which are the molecules of life. DNA, or deoxyribonucleic acid, is the master instruction manual for all living organisms. It's a very long molecule shaped like a twisted ladder known as a double helix. Actually, as you can see in the picture, the DNA double helix contains some main units. What makes up this ladder? Actually, DNA is a polymer, a long chain made of repeating units called nucleotides. Each nucleotide actually has three parts: a sugar, specifically the oxyribose in the DNA molecule, then a phosphate group, and another one is one of four nitrogenous bases, which would be adenine, or A for short, guanine, or G, cytosine, or C, or thymine, or T. The sides of the twisted ladder are formed by the sugar and phosphate groups, which creates a strong sugar phosphate backbone. The rungs of the ladder are formed by nitrogenous bases. A crucial rule governs this structure, which is complementary base pairing rule. According to this rule, adenine always pairs with thymine, which we will have AT bonds, and guanine always pairs with cytosine, which is GC bond. This specific pairing is held together by hydrogen bonds, which act like a perfect fit or a unique Lego piece. This simple rule is the secret to how DNA can be copied so accurately, because we have a specific complementary base pairing. Now let's talk about the DNA close relative, which is the RNA molecule, or ribonucleic acid. While DNA is the long-term storage unit in the nucleus, RNA is a temporary working copy that acts as a messenger. The main differences are that RNA uses a different sugar, which is ribose, and a different base, which is uracil instead of thymine. So, in RNA, adenine pairs with uracil, which we have AU bond. This allows RNA to carry genetic messages from the DNA in the nucleus out to the rest of the cell, where proteins are made. And actually, I have to say, we'll be familiar with other main functions of RNA in the next episodes. Then we want to talk about DNA replication, which is the process of making a perfect copy. The ability to copy DNA is fundamental to life, and every time a cell divides, it needs an exact copy of its DNA. This process is called DNA replication, and it's the masterpiece of biological engineering. Replication is a semi-conservative process. This means each new DNA molecule is a hybrid made of one original parent strand and one newly synthesized daughter strand. This mechanism guarantees that the genetic information is passed down without loss or error. The process involves a team of specialized enzymes. First, the enzyme DNA helicase acts like a zipper. DNA helicase can unzip the double helix by breaking the hydrogen bonds between the base pairs. This creates a Y-shaped structure called a replication fork. Then, in the next step, DNA polymerase takes over. This is the main builder. DNA polymerase can read the original DNA strand and adds new complementary nucleotides, which can build a new strand. It's a highly accurate enzyme with a built-in proofreading function to fix any mistakes. In the next step, because DNA polymerase can only build in one direction, the process creates two different types of new strands, the leading strand and the lagging strand. The leading strand is built continuously in one long piece. However, the lagging strand is built in short segments called Okazaki fragments. So finally, the enzyme called DNA ligase can act like molecular glue, which can join these Okazaki fragments together to form a continuous strand. DNA replication is a complex process that starts at multiple points along the chromosome, forming replication bubbles that eventually merge, ensuring the entire genome is copied efficiently. Then we want to talk about the chromosome structure. So we have this incredibly long DNA molecule. Actually, it seems that there is a fascinating process of packaging. Think of a chromosome as a very tightly wound ball of yarn, where the yarn is your DNA. As you can see in the picture, at the first level of packaging, we have nucleosomes. The first level of packaging is the most fundamental. The DNA double helix is wrapped twice around a group of 8 specialized proteins called histones. This forms a complex known as nucleosomes, often described as beads on a string. Then, in the next step of packaging, we have chromatin fiber. These nucleosomes then coil together into a thicker rope-like structure called a chromatin fiber. In the third step, we would have loops and scaffolds. This fiber is then organized into large loops that are anchored to a central protein framework, which can create a more compact structure. And at the final step, we have chromosomes that are further compacted to form the highly condensed structure of a chromosome, which becomes visible during cell division. And you can see chromosomes under a microscope during cell division. This multi-level packaging is essential for more than just fitting DNA into the nucleus. It also plays a crucial role in gene regulation. When DNA is tightly coiled, the genes in that region are generally turned off, making them inaccessible to the machinery that reads them. When the DNA is more relaxed, those genes can be expressed. This dynamic process of packaging and unpackaging is a fundamental aspect of how our bodies control which genes are active in different cell types. And so, understanding these concepts, which are DNA, RNA structure, replication, and chromosome organization, provide a solid foundation for everything from genetics to medicine. Congratulations, you've made it! Every video you watch is a step forward in mastering genetics. Keep going. Your future expertise is already taking shape.